You will not force
I want to talk about some high-level conceptual things before we get into mathematical modeling. I took this from Ian Stewart's book. It's a 15th century woodcut. And what it says, it gives you an illustration of the fact that the concepts that we use to think about science are often given to us by mathematics, and they limit the science that we can do and understand. In the 15th century, the prevailing view was that God would use only perfect geometric figures to effect natural motion, and so all natural motion had to be resolved in terms of straight lines and circles or segments of circles. So if that's your cognitive stance, if that's your quasi-religious belief, then the trajectory of the cannonball is a straight line followed by a circular segment followed by a straight line downward. That's the best you can do with these concepts. And of course you're going to get a lousy physics out of this, or no physics at all, but that's the limitation of the concept. Now I want to fast forward to the 20th century, and I want to talk about another dominant master concept that I'm going to try to suggest is imposed upon the data, whether the data fit or not. And that is the notion of equilibrium. Now, where do we see the concept of equilibrium? In every single science. You open page 1 of the chemistry book, and they talk about number of chemicals in a box, and they quickly go to chemical equilibrium. We take a physics class and we learn about thermodynamic equilibrium, where the hotter object warms the cooler object until they come to an equilibrium temperature at which T1 equals T2. You take an ecology class and you learn about the carrying capacity of the environment and the concept of ecological equilibrium. And the slow population grows to the carrying capacity. If it were above the carrying capacity, it would shrink to the carrying capacity, and we have the concept of ecological equilibrium. We go take an economics class, and again we see the same thing. We learn to make the sign of the cross, where supply meets demand is the equilibrium price, and we're taught that a free market quickly reaches that equilibrium price. And in physiology, we also are dominated by an equilibrium paradigm, if I can use that word. And that equilibrium paradigm is called homeostasis. And it says that physiological quantities are maintained at constant levels by feedback loops. And in this case, just pull this off the net, as everyone knows or think they know, normal body temperature was 37 degrees, and these are maintained by negative feedback loops to restore normal body temperature. So in addition to the fact of equilibrium, I'd already alluded to the mechanism of equilibrium. The mechanism of equilibrium is negative feedback. Deviation is restored, and we have equilibrium. Probably a very popular example of this in physiology is the feedback loops that control hormone secretion by the target organs of the gonads, the adrenal cortex, the thyroid. All are involved in negative feedback loops, sometimes multiple negative feedback loops. I will stylize it here as a single negative feedback loop, although for research purposes we would want to get a little more complicated. The hypothalamus excites the pituitary excites the gonad, and the gonadal secretion suppresses the hypothalamus. So there is your negative feedback loop. We make a simple 3-variable differential equation. Hypothalamus, pituitary, gonad. This 1 over 1 plus g squared, that's a descending sigmoid, which is the negative feedback. And we simulate this model, and indeed the quantities go to equilibrium as advertised. It's interesting, we see the tracing of the three quantities here, H, P, and G, going to equilibrium. It's kind of interesting to plot that trajectory as a trajectory in 3-dimensional HPG space. And when we do, we see what is called a stable equilibrium point, also known as a point attractor, where no matter where you start the system, except 0, 0, 0, any initial condition anywhere is going to go to the stable equilibrium point, and that means if we perturb off the stable equilibrium point, the system goes back to the stable equilibrium point. And so that is the picture of the homeostatic mechanism. Let's talk quickly about gene regulation, because as you know, genes are the dominant subject in physiology today. P53, the guardian angel gene, the guardian watchman gene, the big anti-cancer gene, is in a negative feedback loop because it induces a protein called MBM2, which in turn inhibits P53. So there is a negative feedback loop in P53. HES1, extremely important protein in morphogenesis, in the laying out of the body plan, somatogenesis. HES1 produces HES1 RNA, which produces HES1 protein, which represses the expression of the HES1 gene. So there's another negative feedback loop. And then, of course, to come back to temperature, temperature is maintained at 37 degrees by a negative feedback loop. So that's a beautiful picture. We have all of these quantities being maintained at steady state levels by negative feedback loops. And it just makes you happy to see this. It's so beautiful. The only problem is it is completely false from beginning to end, and every word that I have said to you in the last 10 minutes is a lie. But normal body temperature is 37 degrees. That's the one physiology fact that everybody thinks they know. Here's the reality. Normal body temperature oscillates. These are student volunteers. Over 4 days, you see day and night in white and gray. And what you see is, number one, there's an oscillation there that is 1 degree Celsius peak to trough. That's a serious oscillation. It seems to be very stable day to day. It has a constant waveform. Notice how it rises and saturates and stays high, then plunges late in the evening and stays low all night. This is a regular, reproducible, physiological phenomenon. And yet everybody here is walking, not everybody here, but in the general public, is walking around with a complete misunderstanding of this. Nobody seems to have noticed, and I do have to say something, because this is a theme that I'm going to start touching on. The data are published in a reputable journal. It's been 50 years, and yet this data is probably new to even most of the people in this audience, let alone the general physiology public. So that's kind of worth remarking, that I have to stand up here and deliver you the news that was delivered in Science 50 years ago. Let's talk about hormone regulation. There's the negative feedback loops. They maintain steady state levels in hormones, except they don't, because hormones are not maintained at steady state levels by anything. Here's a typical luteinizing hormone, an estradiol, over 24 hours in a healthy female. And look at what you see. Number one, there's a big day-night oscillation. During the daytime, LH has these smaller oscillations. At night, they get very large, very well formed, and of lower frequency. So we have oscillations here at a number of time scales, the 24, the 12, and then these three-hour oscillations here. Estradiol, on the other hand, has large 2-hour oscillations during the daytime and then is fairly constant with some noise at nighttime. These are profound oscillations, and in fact, all of the physiological variables of the body oscillate like crazy. Serum potassium, look at that over 4 days. Catecholamines are all undergoing profound oscillations. Let's talk about the P53 negative feedback loop that maintains stability in P53. The group at Harvard Systems Biology had the revolutionary idea that they wouldn't just consult their personal philosophy of equilibrium, they would actually do an experiment and look to see how P53 changed over the course of time. And what they found was clear oscillations in P53 and corresponding oscillations in its inhibitor MDM2. So here's P53 in green and MDM2 in red. That's 29 hours there. And so you see 2 hours of P53, 2 to 3 hours of MDM2. This is an oscillatory system. Let's talk about HES1. So HES1 represses its own transcription, but the result is not constancy. Here is HES1 expression over 12 hours, and you see very clearly a 2-hour oscillation in the expression of HES1, with a corresponding 2-hour oscillation a little bit out of phase with it, as it should be, in HES1 mRNA and HES1 protein, which inhibits the transcription of HES1. We haven't talked yet about NF-kappa B, which is sort of everybody's favorite protein. Group at Caltech looked at NF-kappa B expression in a number of different cell lines, and they found high expression, low expression, high expression over 6 hours. Finally, let me ask you a question, you biologists. What happens when sperm meets egg? The moment of the genesis of life. The answer is intracellular calcium oscillations. That is the first act of morphogenesis in the fertilized embryo, that post-fertilization we begin to get regular oscillations in intracellular calcium, which serve as a kind of a time beater for the subsequent, the clock, to time the subsequent morphogenetic processes. Let me go back to chemical equilibrium for a second, because it's a very powerful concept and we all learned it. In the early 1950s, a Russian chemist named Beluzov had a laboratory model of the Krebs cycle, the reduction of malonic acid by bromate. But it's a model of the Krebs cycle, and he gets up this reaction in his lab, And this is what he observed. He observed oscillations in all the reaction intermediates and products. And that's an awfully good, I mean, that looks like a mathematical model, but that is his chemistry experiment. And so he was very excited. He writes up the paper, sends it to a big Russian chemical journal. Big Russian chemical journal sends it right back to him and says, do not insult us with this. So he gets a friend of his who speaks English, and they rewrite the paper in English, and they send it to Nature. Nature bounces it right back to them, and the editor includes the note from the reviewer, and the note from the reviewer said, we really must develop a form letter to send to these crackpots who think they have invented perpetual motion machines. Now, what's interesting about that is, I was having lunch about 5 years ago with a friend of mine who's a chemistry professor. And we were talking, I mentioned oscillatory chemical reactions. He said, oh, oscillatory chemical reactions, very interesting. I've always wondered about that. How do you make one? And I said, well, the recipes are in the papers. Let's go look up a paper. He says, oh, I got that. Let's go to my lab. He starts grabbing things off his shelf, including concentrated sulfuric acid, so don't try that at home. But he grabs a bunch of things off his shelf, he mixes them up, he gets an oscillatory chemical reaction. So now I want to come back to that point that I raised earlier. The reviewers of Belousov's paper could have done the same thing. They could have grabbed a bunch, this guy's crazy, grabbed a bunch of reagents off the shelf, and they would have seen that he's right. But they didn't do it. And they didn't do it because they didn't need to do it, because they already know that this is impossible. And there is really no point doing the experiment. It's like, you know that pigs don't fly, and you're not going to do an experiment to verify that pigs don't fly, because you know damn well that they don't. And there's an interesting psychiatry there, which is denial behavior, a kind of a mass psychosis that says, I don't want to see the data, I don't want to do the experiments, and we believe in experiments. You know, you have to do the experiment to verify the theory. Nobody did them because of this psychiatric clinging to the concept of equilibrium and damn the data. So this is really a kind of a scientific psychosis of denial of pretty straightforward reality. So let's hold that there, because having established so I take it that I have established the fact of oscillation in spite of all of this denial behavior. Next question, what are the mechanisms of oscillation? So, what are the mechanisms of anything in physiology? It's a funny question. Mechanisms of oscillation. Because in physiology, there's already a view of what is a mechanism. Mechanisms in physiology, according to this view, are the tiny little proteins and the genes and the components of the genes and the subcomponents of the genes. That's the smallest physical and chemical elements of a process. That is the mechanism of the process. And so we see things like the mechanism of the cardiac long QT syndrome, or the mutations here in the cardiac sodium channel. And there are indeed mutations in the cardiac sodium channel, but what do they tell us about what is happening in the heart when you have the arrhythmias that are associated with that gene defect? How does that gene defect surface in the whole heart arrhythmia and in the EKG pattern that we see? And the answer is, we don't know. But we still believe that the mechanism is there in the gene, but we can't really take you from the gene defect to the actual phenomenon itself. So I reject this idea that mechanism and physiology are the tiniest little elements, because they're not really telling us anything. Let's talk about what the mechanism, for example, of oscillation is. Well, that has one paper already put their finger on a key dimension. Oscillatory expression of HES1 regulated by a negative feedback loop. So negative autoregulation of HES1 transcription is what is responsible, in their view, for these oscillations. And keep in mind that we already said that negative feedback explains homeostasis. Now we're hearing that negative feedback explains oscillations. So I'm going to address that in a minute. But in their paper, they lay out this model. We simulated this model, and indeed we saw 2-hour oscillations. The P53-MDM2 system, as described by the Harvard group, they put forward a straightforward 3-variable differential equation model, and we simulate it, and we indeed get the oscillations that they claim would be produced. Let's talk about NF kappa B. NF kappa B is also involved in a negative feedback loop. NF kappa B here is sequestered or locked down by these proteins. An input breaks the lockdown, frees NF kappa B. NF kappa B translates to the nucleus, where it creates more of the proteins that block it. So that is another negative feedback loop, and when you simulate their negative feedback loop, you get oscillations. And indeed, when you knock out the genes for some of the suppressor proteins, you lose the oscillation. Although when you knock out other genes that are less obviously involved in the oscillatory mechanism, the oscillation continues. So here's another point to hold on the other hand, which is negative feedback can create oscillation. And it's probably the paradigm case of this is the predator-prey equations, which surfaced in the 19-teens, 100 years ago. Imagine a population of sharks and tuna, and the tuna population increases the shark population by feeding it, and the shark population decreases the tuna population by eating it. And the result of a simple model produces oscillations in the shark populations and the tuna populations. Now, we did a little experiment with this model. We asked ourselves, suppose we have the view that we want more tuna and we don't like sharks and we want to free the tuna. So we do the experiment of simply removing sharks. So we institute a shark removal program right there, and the population of sharks declines. And then the population of tuna rebounds to a higher level, as hoped for. But when that happens, the shark population also recovers to a higher level than before. So the net effect of removing sharks is to increase the peak number of sharks afterwards. Moral of story, complex systems defeat naive interventions. You just can't go in there and say, I want more tuna and fewer sharks, and so I'm just going to lower the shark population, and there you go. Because the system will not stay there. from that point of view, it becomes difficult to understand statements like this, which I pulled out of a paper. We're going to unleash P53 by blocking MDM2. And we heard this from a speaker a couple of weeks ago. I pulled this out of a paper. Everybody's trying to unleash P53 by blocking MDM2. And that is exactly increasing the tuna population by blocking the sharks. And you will get a higher peak, but you'll also get a higher peak of MDM2. You will change the frequency. You will do all kinds of stuff. You can't simply look at this as, I'm going to take away the inhibitor and unleash the activator. That's bar stool thinking. That's pub stool thinking that is really not worthy of this kind of phenomenon. So, let's go back to our question. Does negative feedback promote homeostasis? Or does negative feedback promote oscillation? We've heard both views. There's an answer. And the answer is the Hopf bifurcation theorem. So here's an example. Let's take the classic negative feedback of a thermostat and a heater. The thermostat turns on the heater puts out heat, which turns off the thermostat. There's your negative feedback. We make a simple model of this. T is the thermostat state, H is the heater state. We make a differential equation, and in this differential equation, the thermostat is going to respond to the heater. Notice 1 over 1 plus h to the n, that's a downgoing sigmoid. And we also have a time delay, tau, here. And h tau is simply h tau seconds ago, the time delayed value of h, because that's what the thermostat sees. It doesn't see what the heater is putting out right now. It sees what the heater was putting out 20 seconds ago, 30 seconds ago, depending upon the circulation of air in the room and the distance from the heater to the thermostat. So now we have this model that has two parameters, the time delay tau and this exponent n, which governs the steepness of the negative feedback. Low n is gentle feedback, high n is steep feedback. And now we apply the Hopf bifurcation theorem to this model, and this is what emerges. That we can look at the two parameters, and we can make that two-parameter diagram, and the theorem says that there's a hyperbola there, and if the product of the slope of the negative feedback and the time delay is greater than a certain constant, then you're in oscillation. And if the product of them is below that, then you're in the equilibrium. So there is an exact answer to our question. Negative feedback loops promote homeostasis when the slope is mild and the time delays are small. But whereas if the feedback is steep and the time delays are significant, you are going to get oscillation. So let's talk about, for example, slope of the negative feedback. We talked about the hypothalamic pituitary gonadal system, and you saw that model, and you see that 2 there. That 2 is a gently sloping sigmoid. 1 over 1 plus x squared is a gently sloping sigmoid. And when we do this model, we indeed get equilibrium. What happens when we increase the steepness of that, the harshness of the feedback, or the sensitivity of the hypothalamus? And the answer is the system goes into oscillation. So just that same model with n equals 10 instead of 2 gives us stable oscillations in all three quantities. And now if we do that technique of plotting the three variables as a trajectory in HPG space, we get that. And what that is telling us, here's HPG space. This is a trajectory in 3 dimensions. If we start at very high values, we spiral into a red loop. If we start inside very low values, we spiral out to the red loop. All non-zero initial conditions in this system go to the red loop. This is a new object. This is called a limit cycle attractor. And what it represents is a stable oscillation. Because wherever you are, you're going to go to the red loop. That's the dynamics of the system. Notice that all of the oscillatory systems we learned about in physics, the harmonic oscillator, the pendulum, the spring, don't have this property. If you pull the spring out to a larger initial condition, it yo-yos back and forth to the larger initial condition forever. But if this is body temperature and you have a fever one day, you would love to be able to go back to your normal body temperature oscillation and not stay in a large amplitude oscillation forever, which is what the models of physics predict. So this limit cycle attractor is a very important new concept, and this is the mathematical model for physiological oscillations. Very pretty work by late Julian Lewis and Nick Monk also stressed the importance of time delays in negative feedback loops, that transcriptional delays provide sufficient time delay to get the time delay plus negative feedback mechanism going, that they can therefore account for oscillations. The Nobel Prize 2 years ago was given to Hall, Rosbash, and Young. And if you ask most people, they'll say, oh yes, they're the ones who discovered the genes, period and timeless, and those are the genes that govern the circadian rhythm. It's a mistake. The period and timeless, the genes that are involved in the circadian rhythm, have been known for 20 years, not due to them. What they did, which was truly interesting, was they deduced the mechanism of the oscillation that is the circadian rhythm. And that's not something that you do by staring at the gene or its subcomponents. You have to understand, this is all their drawings and their words, that period and timeless produce products that come back and inhibit their own transcription. In both cases, there's the inhibition. And then they make the point that there's autoregulatory feedback loops, that is negative feedback, and oscillations are achieved by delaying various steps in the network. For example, period is retarded in the cytoplasm by phosphorylation. So this is a mechanism of oscillation. You could make the mistake of saying, well, I'm a physiologist. I certainly recognize the word phosphorylation. We have a lot of phosphorylation going on in a lot of our systems. And so the mechanism of the oscillation here is phosphorylation. But that's really a mistake, because phosphorylation in 99 other systems does not produce oscillation. The important mechanism here is time delays in a negative feedback loop. And anything, any molecular changes whatsoever that are going to produce oscillation are going to produce it by either increasing the slope of the negative feedback or increasing the time delay. All wannabe mechanisms must go through one of those two mechanistic factors. Talking about time delays, let's talk about economic equilibrium. We can make a very simple model where the change in price is equal to the total money available on demand minus the total amount of money available for supply. When demand is greater than supply, P prime is positive and the price goes up. When supply is greater than demand, this term is negative and P prime is negative and the price goes down. And we make a simple model of this as a positive exponential and a negative exponential. And we simulate that model, and Lord, if it doesn't go right to equilibrium with supply equal to demand and the price is stable and everything we learned in economics is true. Except Mike Mackey, who's at McGill, should have 3 Nobel Prizes in economics because Mike discovered an unbelievable fact, which is it takes time to grow corn. It takes time to make an automobile. So the adjustment of supply to price is not instantaneous. There are time delays in that process. So this is genius. It takes time to grow corn, folks. We make that change in the standard equilibrium model. We have supply is now reacting not to price, but price tau time units ago. And we run that model, and we get big time oscillations in supply and demand and price. Now, I have to say something about this. I'm sorry, I'm going to be very unpopular. What the heck is going on here? 100 years, well, 80 years of economics students have been taught equilibrium, supply and demand, and only Mike Mackey from McGill discovered that it takes time to grow corn? This did not occur to the Nobel Prize winners of the Chicago School of Economics? What's up with that? What do you want to say about that? What is their mental state? That this is a revelatory discovery. So what I want to say is the Hopf bifurcation theorem gives us the mechanism of oscillation. It's the product of sensitivity and time delay, and anything, I will say this one more time, any wannabe cause that wants to promote or abolish oscillation must act through one of these two factors. This is the true mechanism. Let me go on. I hope I've argued that oscillation is a widespread phenomenon. Why? Why do we have all this oscillation? What is the teleological function of all of this? So there was an absolutely stunningly beautiful paper which got very nicely published in Science, which I never see referred to because it's unpleasant. And they're looking at yeast metabolism. And here's oxygen pressure, and as you see, it oscillates in yeast. And then these people, or rather their graduate students and postdocs, looked at 6,209 open reading frames over time. And this is an overplot of 6,209 time tracings. And you'd expect to see total junk, but you see definite form here in this overplot, and you also see that things are coordinated with O2 pressure in the metabolic oscillation, that these are all happening at this phase. So then they looked at the 6,209 genes, and they were actually able to segregate them into 3 groups. So this is an overplot of all of those genes on the top line. This is an overplot of all of these genes. This is an overplot of all of these genes. Notice the consistency in the waveforms, which is really remarkable. And then they're able to say that these 3 groups, which they give names to, the oxidative group, the reductive building group, and the reductive charging group, these succeed each other in regular succession, the red group, the green group, the blue group, and the red group, the green group, and the blue group all occupy preferred places in the respiratory cycle. So they coined the term temporal compartmentalization. What do you do with incompatible processes? Some processes require high pH, other processes require low pH. Some processes are oxidative, others are reductive. How do you reconcile that? Well, there are 2 ways. One is spatial segregation, but the other is temporal segregation. You take care of different aspects of your business at different times. And the incompatible conditions, you merely cycle through them. So the picture that emerges, and this was drawn very clearly by the News and Views article about this paper, is a very pretty picture of physiology. And this is what I'm suggesting should replace this homeostasis. It's saying that out in the periphery, liver, lungs, you have all of these peripheral oscillators. And core body temperature, which of course speaks to all the organs of the body, that is the one ring to rule them all. And it synchronizes and governs those peripheral oscillations, which are then entrained to the day-night cycle by the suprachiasmatic nucleus and the oscillations in the suprachiasmatic nucleus. But the day-night cycle and the suprachiasmatic nucleus are not the causes of the oscillation. They are merely synchronized to that by these peripheral oscillators as governed by core body temperature. Again, functions of oscillation. This is a very pretty paper going back to the HES1 oscillations, which we saw, that says that here's the HES1 protein oscillations. Different cell commitment fates are seen at the up phase as opposed to the down phase. High HES1 means notch is off, and you get a mesodermal fate for these stem cells. Whereas low HES1 means that its inhibitor, or the thing that it inhibits, the notch signal, is on, and you get a different, you get the neuroectodermal fate. So the cycling produces different commitments of cell lineage. I want to talk now about an example not of good oscillations, but of bad oscillations. And how do we use the Hopf bifurcation criterion, in this case not to create oscillations, but to abolish them when they're unwanted, when they're pathological. So here is a simulation of ventricular fibrillation. As Raj correctly said, the leading cause of sudden cardiac death. Here's a normal beat. Follow the bouncing ball, we get another normal beat. And now something bad is about to happen, an ectopic beat right there. And that ectopic beat is now going to produce a stable, unfortunately stable, chaotic state of electrical conduction, which is what ventricular fibrillation is. This is where they yell clear on the medical shows and they apply the paddle, which is, of course, a massive intervention. This is VF. So what's going on in the tissue in VF? It was funny, for 100 years it was described in Victorian literary terminology, frenzied and incoherent contraction of a bag of worms. I want to say this is the leading cardiac killer, and that's the best we can do, is describe it as a frenzied and incoordinated contraction. So it wasn't until the 80s and the 90s and the advent of computerized mapping systems, this is a 30 by 30 computerized electrode system, that scientists were able to say what the heck is going on there, which is multiple waves of electrical excitation colliding with each other and extinguishing, and then new waves, daughter waves, being formed by a process of wave break. So that's what the mapping systems can tell you. So then the next question is, what causes breakup? Cardiologists were 100% clear that what causes breakup is external heterogeneities, shown here by the black triangle. And there are plenty of them. You have necrotic regions, fibrotic regions, ischemic regions, lots and lots of heterogeneity. And the idea was, as when I was starting out, a cardiologist said to me, son, it's just like a wave at the beach. It hits the rock and it divides into two. So this is a form of explanation that says explain things like breakup by appeal to externalities. You bump into a rock. Dynamicists in the 90s realized that there was another mechanism for breakup. And I'm going to show it to you. Here is a 600 by 600 grid of cardiac cells, a very simple 2D model of cardiac tissue. We're going to send a wave from left to right. The heart is going to recover. And now we're going to put that ectopic beat in there. And there you see the genesis of a spiral wave. And you see that that spiral wave is inherently unstable and is breaking up of its own dynamics in mathematically homogeneous tissue. So don't talk to me about external breakers. The breaker here is internal. And psychiatrists like to talk about internal locus of causality and external locus of causality. And if you have an external locus of causality, that's unhealthy. And unfortunately, the external locus of causality view seems to dominate in a lot of the sciences. Asked why breakup, you look for externalities. But here's a dynamical explanation of breakup. Here's a cardiac action potential produced by a stimulus. During the diastolic interval, the ions are all being pumped back out of the cell and into the cell. And you wait a nice long diastolic interval and you give a second stimulus and you get an action potential identical to the first. But if you begin to encroach on the diastolic interval, you're going to get, because the process of restoring the ions hasn't completed, you're going to get a stunted action potential. The red action potential duration is shorter than the white. If you encroach further on the diastolic interval, the blue action potential is even shorter than the red. And if you encroach majorly on the diastolic interval, the green action potential is so stunted that it does not have the force to propagate. And that is going to be an engine of wave break right there, that very, very short, weak action potential. So we make a curve of the diastolic interval on the x-axis and the following action potential duration on the y-axis, and that is called the action potential duration restitution curve. And the hypothesis is that a steeply sloped restitution curve is going to produce violent oscillations in action potential duration. Because suppose now we force the heart from the pacemaker at a nice long cycle length, we get this periodic train of action potentials. But now, if we start to pace fast, the long action potential gives us no diastolic interval before we ask for the second one, and that's going to mean a stunted second one, which is going to give us a long diastolic interval, which is going to give us a long third one. So we have a negative feedback. Long action potential produces next one short. Short action potential produces next one long. There is a negative feedback loop with a time delay. And those oscillations in action potential duration, when they become violent, large amplitude, the very short ones are going to be too short to propagate, and there is your engine of wave break. And in fact, in general, steeply sloped relations are input-output relations. Steeply sloped input-output relations are magnifiers of differences. You put a difference delta x into a steeply sloped relation, and delta y is bigger than delta x. In other words, the small difference got magnified. If you have a shallowly sloped relation, slope less than 1, then the same difference gets translated on the next beat to a smaller difference. These relationships heal differences. These relationships magnify differences. So we thought, well, that's interesting. Let's try this in a cube of tissue model. We tried a couple of drugs, that is, we type in different ideas for drugs. And what you see here is two steeply sloped feedback relations, one with a long action potential and the other with a shorter one, but it doesn't matter. You get breakup and VF in either of B or C. Now, if you flatten that relationship, reduce the slope, now the spiral wave remains intact. And pharmacologists like to talk about, we talk about anti-arrhythmic pharmacology, and one of the big categories is class 3 potassium channel blockers. So this is class 3 action here in B, and this is anti-class 3 action here in C, but you see that it don't matter none. What really matters is the slope, not class 3 or not class 3, but did you flatten the slope or did you not? That's the deciding factor. So we figured, let's try this in a whole heart model. So here's VF in a whole heart model. You see the spiral wave immediately broke up into the multi-wave fibrillation. We put in a model drug that lowered the slope, and the spiral wave does not break up. So we started showing these, and cardiologist said, well, son, the heart is much more complicated than your mathematical models. There are all of these externalities, ischemia, fibrosis, necrosis, what have you. So one of our group, TJ Wu, is now chief of cardiology at National Taiwan, decided to do an experiment in a rabbit heart. So here is an isolated Langendorf rabbit heart, imaged with a voltage-sensitive dye, so you can see the voltage on the whole surface of the heart. And here is fibrillation in a rabbit heart. Red is depolarization. You see the waves are coming from the left. Now they're not. Now they're going around and around. Then that's going to stop, and they're going to start coming in from the top right for a while. This is fibrillation. When we administer, TJ and his advisor, in consultation with us, found this drug D600, which is a fast-acting calcium channel blocker. This is a heavy calcium channel blocker. You wouldn't want to use this clinically. But this heavy calcium channel blocker, in a dose-dependent manner, reduces the slope of that curve. So we gave a nice heavy dose of it, lowered the slope of that curve, and the induced spiral wave remains intact. In spite of necrosis, fibrosis, anatomical heterogeneity, all of those externalities that were supposed to be the causality of breakup didn't have that effect. So this is a further argument for understanding mechanisms. And I want to close. Raj mentioned the most sacred name of them all, and I'm going to get to that. I want to talk just for a few minutes about spatial pattern formation. And these are, of course, not temporal oscillations. These are spatial oscillations. And we want to ask the same question: where do spatial oscillations come from? For example, here's segmentation in Drosophila. Pre-segmentation, embryo is pretty homogeneous, except for the nucleus there. And post-segmentation, look at this pattern. Very regular. Notice that the curvature increases of the segments as the curvature of the embryo increases towards the ends. How do we explain the emergence of a pattern like that? The person who answered that question is Alan Turing. And Turing does not get enough credit for this. I think people think, oh, he just looked at plants and he liked to explain animal coat patterns, and isn't that cute? And where does the tiger get its spots? I think that's a huge disservice to Turing, because his real contribution is fundamental and gigantic. What he really is, this is the famous paper, chemical basis of morphogenesis. A system of chemical substances called morphogens, and he is coining this term in this paper. This is the first use of the concept of morphogen. Reacting and diffusing is adequate to account for the main phenomena of morphogenesis. Now look at what he says here. Such a system, although it may originally be quite homogeneous, may develop a pattern or structure due to an instability of the homogeneous equilibrium. In other words, the equilibrium is unstable and the system will go into spatial oscillations. What is his model? It requires two things. It requires short-range activation, positive feedback, and long-range negative feedback. So the short-range positive feedback builds up the pattern, But if there were no inhibitor, that would simply then take over the whole space. You'd have one huge spot or one huge stripe. The role of the long-range inhibition is to sculpt that pattern, produce voids in between the maxima. And that's where pattern is going to come from. We make a model of this with two chemicals, an activator and an inhibitor. The activator activates the inhibitor inhibits the activator. They both diffuse, but the key is the inhibitor diffuses faster than the activator. Turing himself used the story of, think of these as little fires, and think of these as little firefighters. Now, if the fire progression, if the diffusion of the flame, the diffusion of the fire, is faster than the firefighters can run, then you're going to get one big fire. But if the firefighters can run faster than the fire, then they can aggregate into little firefighting units and create a fire-free zone. And you're now going to begin to get spotty expression of fire and firefighters. We put this into a partial differential equation of the reaction diffusion type. And there is a Turing bifurcation, which is really the Hopf bifurcation in space. Just like the Hopf bifurcation we saw before was in time, now the Turing bifurcation is sometimes called the Hopf-Turing bifurcation because the Turing bifurcation is in space. And it says that for low diffusion rates, notice that the axis here is space, not time. For low diffusion rates, the homogeneous equilibrium is stable. But for rapid diffusion of the inhibitor, the homogeneous equilibrium is unstable and produces a sinusoidal spatial pattern. There's an example of the simulation of a reaction diffusion equation in an oval-shaped domain. Notice how many of the features of segmentation are captured by this incredibly simple model. For example, the fact that the boundaries of the segments are everywhere perpendicular to the outer surface is captured without further effort. Emerges, which is the right word there, emerges directly from the reaction diffusion model. So this kind of explanation is very, very powerful. And I've gotten into a lot of unpleasant discussions with people over this. And they say, well, this is a lot of crap, what one Nobel Prize winner said to me. It's the biggest piece of crap I ever heard. And I said, thank you, sir. Thank you for sharing that. Why? And he says, well, everyone knows that segmentation is carried out by homeobox genes, which are bicoid expressed in this, and there's a gradient of that, and gives me a whole molecular biology piece of hand-waving. And doesn't really answer the question, where does the pattern come from? I'm going to give you one more example, and then I'll let you go. There was an incredibly beautiful paper 10 years ago in Nature. They looked at the branching program of the lung. The lung, obviously, develops embryologically by branching. And these people and their graduate students, detailed 3 different types of branching in the lung. Side branching in blue, orthogonal rotation of the branching plane in red, and planar branching tip splitting in green. I won't have a chance to do the blue or the red, but I do want to address myself to the green, to the tip splitting. Here's tip splitting. Here, by the way, is orthogonal rotation of the branching plane. The first bifurcation in the anatomical sense is in the lateral medial plane, and then the next one is in the anterior-posteral plane. So they have this beautiful documentation of these forms of branching in the lung. But what is the mechanism? And here one is profoundly disappointed. All they can say is each mode of branching has a genetically encoded subroutine. And it will be particularly important to identify the genes that underlie the periodicity generator, the bifurcator, and the rotator. There was a News and Views about this paper in which the author chose an extremely unfortunate metaphor of the master and the slaves, which is unfortunate not just for its social connotation, but its bad biology. I'm not just saying it's offensive socially, it's bad biology. And what are they saying here? They're saying, how does bifurcation happen? There's a bifurcator gene. What causes orthogonal rotation? There's an orthogonal rotation gene. What determines the length of the segments? There's a periodicity gene. This is hand-waving. How is this different from saying, why does bifurcation happen? It pleases God to have bifurcation. How does orthogonal rotation? God wills orthogonal rotation. This is nothing more than that. It's the brainless postulation of a gene for every possible condition. If you're wearing a blue sweater, there must be a gene for wearing blue sweaters. This is very unsatisfactory. This is bad science. It's beautiful description, and when they go for the mechanisms, all they can say is, genes make everything happen, so there must be a gene making each of these things happen. Period. That's all that's going on there. So we thought, well, maybe we can do better. We went to an old model, 40 years old, by Meinhardt, which postulates an activator chemical, an inhibitor chemical, a substrate chemical, and a marker for cell commitment. We simulated this model, and tip splitting, and also the other two, which I don't have time to tell you about, but all three modes emerge from that differential equation model with no genes at all. In particular, what causes tip splitting? Here, the activator arises. The activator gives rise to the inhibitor after a certain time delay. The inhibitor then rises and provides the inhibition that splits the peak of the activator. Having split the peak of the activator, the peaks now amplify, and you just produced tip splitting. So in that way, we were actually able to replicate all of the phenomena and give it a very different kind of explanation for these branching modes than simply postulating a gene. So my final slide quotes two of our best mathematical biologists here in town, Dennis and Phil Miney. And look at what they both used the Humpty Dumpty metaphor, which I think is cute. Dennis says, if you look at molecular biology as breaking Humpty Dumpty into many pieces, we need to start, and I totally agree with this, we have been breaking Humpty Dumpty into lots of pieces. I'm tired of seeing the little pieces, and I would like to see somebody start to put them together. And as Phil correctly points out, the tool that will put those pieces together is mathematical modeling. Thank you.